Good morning to everyone. Before starting the webinar, I request everyone to remain muted, both audio and video, throughout the entire session. So we have an eminent scientist with us. We request everyone to maintain decorum throughout the session. If you have any questions to ask, kindly write it down on the chat box. It will be read out by the host after the presentation to the speakers. I, Dilshad Begum, Assistant Professor, Department of Biochemistry, Indian Academy Degree College, cordially welcome all the participants to an international webinar on Trends in Biochemical Research organized by Department of Biochemistry, Indian Academy Degree College, Autonomous. Biochemistry is the branch of science that explores the chemical process within and related to living organisms. By using chemical knowledge and techniques, biochemists can understand and solve biological problems. Much of the spectacular progress in biochemistry research, whose primary goal is understanding of the fundamental working of living things. With this in mind, we have two talks from eminent scientists from USA on cardiovascular interventions by Dr. Prakash Dhanpatar, University of Iowa, USA, and understanding synapse development using Drosophila Neuromuscular Junction by Dr. Soumitra Chowdhury, National Institutes of Health, USA. Now I take this opportunity to welcome our beloved principal, Dr. E. Jerome Xavier, who is always a backbone for conducting such events. I welcome you, sir. Before starting the scientific sessions, I request Principal Sir to deliver the welcome address. So kindly deliver.
Dear resourceful persons, Dr. Prakash and Dr. Soumitra, dear participants, faculty members, research scholars, students from all over Karnataka and India. At the outset, I would like to extend a very warm welcome to each one of you, very especially to our renowned scholars and scientists, Dr. Prakash and Dr. Soumitra, for this fantastic webinar on trends in biochemical research. I was very glad to see the topic because both the resource persons are going to deal with two most important physiological aspects of human and in a very special manner. These are all not only physiologically important, but more so for the overall personality of every individual's mind and heart, brain and heart. We know one of the mysteries in the study of human beings is the study on brain, and the other one is the study on heart. Besides the physiological aspect that controls the heart, we know heart is a seat of knowledge, affection, emotion, and love. While we always consider brain as a seat of wisdom, knowledge, understanding, reason, and so on. Although there are thousands and thousands of volumes written on both and research being carried out both on heart as well as on mind or brain, we all know it is not sufficient. There are areas wherein we haven't discovered, particularly when it comes to brain. Almost every month and every year we find new research articles being published on the study of brain. And from studying brain, now they are moved into artificial intelligence. So soon we will find the technology and the physiology might be combined together to give us new information, new knowledge, a new way of thinking, not only our life, but also our relationship to the environment and particularly to the technology. I'm sure the speakers with their excellent record in research and research publications will be able to enlighten us, although it is a limited amount of time, still I'm confident they'll be able to inspire us, enlighten us, and give new perspectives on these two topics, brain and heart. Once again, Dr. Prakash and Dr. Saumitra, on behalf of the management, principal, and the faculty members of Indian Academy Degree College Autonomous Bangalore, I extend a most hearty welcome. And over to you, the resource persons. Thank you very much for accepting our invitation and being with us this morning. Thank you.
Good morning, everybody. I am Professor Pushpa Reddy from Department of Biochemistry, Indian Academy Degree College, Bangalore. I feel proud to introduce our outstanding alumni, Dr. Soumitra De Chaudhary, as a resource person for this international webinar, Trends in Biochemical Research. To tell you in brief, Dr. Soumitra obtained his master's in MSc Biochemistry from Indian Academy Degree College, Bangalore. And further on, he obtained his doctoral from Indian Institute of Science Education, Bhopal. Subsequently, he carried on his postdoctoral research at National Institute of Health, US. He has very good publications in PubMed. His career goals include understanding the molecular mechanisms of synaptic development and extend the knowledge to a mechanistic understanding of neurological disorders. His PhD training started with presynaptic studies, and later he continued with his postsynaptic studies research in postdoc research. Dr. Soumitra is currently working as a visiting postdoctoral fellow in National Institute of Health Center on Compulsive Behaviors. Now, may I request Dr. Soumitra to present his knowledge on the topic Understanding Synapse Development using the Drosophila Neuromuscular Junction, an emerging concept in biochemical research. So it's over to Soumitra.
Okay. Thank you so much. Am I audible?
Yeah, you're
Okay.
audible.
So first of all, I'd like to thank Indian Academy Degree College for this invitation. And it's really an honor and privilege to be presenting in the same college from where I passed out. And what's really nice is the kind of work that I'm doing is partly also because of the motivation that I got during my master's. And if there are master students around, you probably a course in your semester called Molecular Physiology. And that was one course that fascinated me so much that I kind of tried to take that as my research study. And so quite a lot of the thing that I'll be showing you now involves a lot of signaling, which is something that I first came across during the molecular physiology classes. Again, I'd like to thank the principal and the faculty of the Department of Biochemistry for this invitation. And I think it's an absolute honor to be presenting my work. So let's get started. All right. I think I have to share my screen. Okay. So I think my screen is visible.
Yes.
Okay. So all right. So yeah. So today what I'm going to do is talk to you about understanding synapse development using the Drosophila neuromuscular junction. And the image that you see here is that of Drosophila neuromuscular junction, and you see those rounded structures. That is where the nerve terminals are. It's called synaptic boutons. So I'll be coming to that in my later slides. But before we really get going, let me begin with something very simple. So I believe we've all seen a relay race. For that matter, some of us may have even participated in one. So the point of showing this slide or trying to tell you about relay race is that there is something which is really happening here, and that is uh, the baton is being passed from one person to the other. And I'm using the baton here as an analog for information exchange. Now, the question is, where in the body does this kind of information exchange actually happen? And undoubtedly, that happens in the brains, where you've got 100 billion neurons and each of them forming like 1,000 synapses. And so total number of synapses would be 100 trillion. And each of these synapses work by exchanging information among themselves. So you can see here that there are two neurons, one passes the information and one receives it. So in every context, we talk of that as a presynaptic neuron, which passes the information and that is received by a postsynaptic neuron. Now, I've told you that we can consider this as a baton that is being passed from one person to the other. Of course, the only difference is that instead of the baton that is passed from, say, the presynapse to the postsynapse, we have something called as synaptic vesicles, which are filled with chemicals called as neurotransmitters. And these neurotransmitters then are released in these minute 20 nanometer distance cleft called as the synaptic cleft. And then these neurotransmitters then bind to the receptors which are present in the postsynapse. And then the information or the signal is basically transduced downstream. Now, I've been mostly interested in understanding the different molecules that are present in the presynapse, or for that matter, even the postsynapse, and how these molecules interact with one another, how they formed, how they form modules, which kind of help in the information exchange between the two neurons. And the way to kind of visualize these protein molecules or modules is that you can think that they are individually, they are kind of pieces of the puzzle. And when you put together this puzzle, you have a functional system. And then there is a lot of signaling that is also happening because of these modules. So I told you that this is what really happens in the brain, but then the question is, why should we really care? What is the need to really understand the synaptic processes or the information exchange, the synaptic transmission that is happening in the brain? Well, as it turns out, if some of these processes go wrong, you have disorders like schizophrenia and autism, and these are just to name a few. And so to study these, what I use is I use the fruit fly, that is a Drosophila melanogaster. And the advantage of using fruit flies is that they are pretty much similar to humans. Um, they have a brain, which is pretty much complex. They have something called as the ventral nerve cord, which is functionally equivalent to the spinal cord that we have. And they also have these segmental nerves, which we can then study. And also, flies are very easy to rear in the lab. They have a very short developmental cycle, and basically within a span of a week, you have an adult coming from an embryo. So this whole developmental cycle is pretty short, and that makes them a very suitable organism to work with in the lab. And to add to that, we've got a tremendous array of genetic tools available with us, with which we can perform all sets of genetic manipulation and study the function of a particular gene. And recently, what has evolved is that we've got wonderful databases like OMIM, that is the Online Mendelian Inheritance in Man. You have got the Undiagnosed Disease Network, UDN, and websites like Marvel. So these are the websites that coordinate or have data, patient data, and you can always look back at these particular databases, find the gene, find its homologue in flies, and then try to mutate those particular genes and see what is the phenotype that you can actually get. And what's even more important is that you can also find other fellow researchers who are working on similar proteins. You can also find clinicians who are working with patients. So these resources are of great importance because if you have a clinician who's working on a similar kind of a problem, you can always collaborate with the clinician and then, of course, extend your basic study in flies to the patient data and then understand the function of the gene in greater details. So, and also I would like to take this opportunity here to do a little bit of publicity for this website, which is Drosophila for Schools. So if any of the students are really interested in understanding how the fruit flies are used in research and what are the similarities that they have with humans and what are the resources that are available, the kind of genetics that one can do with flies, this website is really a great place to start with. And it's a basic thing and I can understand a lot of things. So this is something that I'd just like to advocate. All right, so now coming to the model system that we use, that is the Drosophila neuromuscular junction. Um, the one in the top, the image is that of Drosophila larvae, but this is that of a first instar larvae. And then you have the blown up one. So this is the third instar. And you would appreciate that within a span of like 48 hours, the larva grows in size from the first instar to the third instar. And this growth is like 100 folds. So there is an increase in the muscle area 100 times. And to compensate or to coordinate with this 100-fold increase in the muscle area, you have the neurons also correspondingly growing and occupying or innervating these muscles. And since I said like this growth takes place in like 48 hours, so this is a wonderful model to study the developmental functions or developmental uh, to understand the function of a gene that could play an important part during development. And so the system that we use in the lab is that of a third instar larvae. So this is how a larvae looks like. And you can easily make this preparation of fillet by cutting along the dorsal line. So what happens when you cut this off is you kind of expose the larvae, you expose the ventral nerve cord. And then what is shown in the right is an immunohistochemical preparation. So you can basically open the larva and stain it for different markers, and then you're able to see the terminals. So and the other advantage of using Drosophila larvae is that you can appreciate that these segments are like arranged in a very stereotypical pattern. You have the same muscles on either side. So each side is called a hemisegment. They're divided into 7 different abdominal segments, A1 through A7. Um, and so you have the same set of muscles in the different segments, and you can always come back to the same muscle if you want to study an animal life. So so what is shown here is neuromuscular junction, that is the connection or the contact between a motor neuron and the muscle fibers. So you have the ventral nerve cord here, it is depicted in the cartoon. So you have the ventral ganglia here, and that's like the central nervous system. And you have the axons being projected from the ventral nerve cord, and they come out and have a contact with the muscle. And if you see in the bottom panel, you can see those rounded structures. So these are the synaptic boutons or the nerve terminals. So this is exactly uh, the business end of the nerve, and all the activity that I was talking to you about previously about the information exchange. This is the place where this actually happens. So if you kind of look in further detail, so now this is just one of those rounded structures that I showed you previously. And you see 2 different proteins. One labels the presynapse and the other the postsynapse. So the presynapse here is labeled with a protein that is called Brook pilot. It's an active zone protein. So that is at the presynaptic side. And you have something shown in the red that is the receptors, so in the postsynaptic side. So, so this is a confocal image. And if you further go to the details that is shown here using electron microscopy, you can see the magenta here. That is the active, there's a presynapse with the active zone. The rounded structures that you see here are the synaptic vesicles. So they contain the neurotransmitters. And the dark structure that you see here, which is in the form of a T bar, is the active zone. And this is exactly what is shown in this image in the green. So this is the the part in the green. And this is a region where all these synaptic vesicles come, dock, and fuse, and they release the neurotransmitters. And when these neurotransmitters get released, they bind to these receptors, which are shown here in red. So they're again dense, and they're shown in red. And this is on the muscle side for the neuromuscular junction preparation. And you can see a very thin gap, and basically you can see two membrane structures. So this is the 20 nanometer gap or the synaptic cleft that I was talking about. So just to put things into perspective, what you see here in magenta is the presynapse. What you see here is rounded structures are the synaptic vesicles. This small gap is the synaptic cleft, and the green structure that you see here is the postsynapse. So now you've got a fair bit of an idea of how the Drosophila NMJ actually looks like. So with this now, I'll just get on with the main part of my talk. So I've kind of divided the talk into two parts. And what I'm going to do today is mostly discuss my PhD work that was done in IISER Bhopal. And the main, my thesis work was on AP2 adapter and endocytosis. So what I'm going to talk to you about is the synapse morphology and function, and what AP2 adapter does in clathrin-mediated endocytosis, and what happens if you deplete the AP2 adapter complex, or if you perturb the function of AP2 adapter. So what really happens to the neuromuscular junction? And the second part of this AP2 adapter and endocytosis, I'll be talking to you about the various signaling pathways that is affected when you have a mutant form of the complex. And towards the later part, I'll talk to you about a disease called amyotrophic lateral sclerosis, which is also called as the Lou Gehrig's disease, and a particular gene that is involved in ALS, that is Senataxin. And our studies with the human form of Senataxin uh, and our studies on like what the kind of results that we got expressing the different variants of this in Drosophila motor neurons, and what are the conclusions that we could draw. So as I said, the neurons are highly polarized. You have a cell body, you have a long axon, and then you have the synaptic terminal or the synaptic bouton. And now this is the business end of the nerve terminal, because all the vesicles are basically released into the synaptic cleft from this side, and then there are receptors here which take up the chemicals, and then of course the signal is processed downstream. Now, if you zoom in and try to understand what really happens here, is that you've got these synaptic vesicles which are filled with neurotransmitters, and when there is an action potential that comes in, you have calcium coming in through the calcium channels, and that leads to the fusion of these synaptic vesicles with the presynaptic plasma membrane, which is mediated by the snare complexes. So that leads to the release of these neurotransmitters into the synaptic cleft, which then get attached to the neurotransmitter receptors, and then of course the signal is processed downstream. Now there is a fundamental problem out here, that is, once these synaptic vesicles fuse, they essentially add to the membrane of the presynaptic space. So this is basically a bilayered membrane structure that comes and fuses with this membrane. And so you basically add this extra membrane. So if you don't recycle these membrane back, essentially what happens is that you're going to have a bulging membrane, and that will not further allow any more fusion. And a physiological consequence of that would be paralysis. So what are the ways by which you can basically recycle these membranous structures? So the way this happens is by endocytosis. So you have a clathrin-mediated endocytosis taking place, and this is the clathrin coat that forms around these membranous structures. Then you have a GTPase called dynamine that comes and cuts this vesicle off, and then you have the vesicle being regenerated again. The clathrin coat comes off, and then of course you refill this vesicle with neurotransmitters, and these cycle continues. So this is basically called the synaptic vesicle cycle, and this cycle then actually continues. So one of the major modes of this mechanism of endocytosis is mediated by clathrin, and AP2, of course, is the adapter complex that mediates this process. But then there are other modes of endocytosis that also happens at the synapses, and I'll come back to that later. So when I started my PhD, I started with like this three major questions. So the first question was, what is the role of the AP2 complex in presynaptic vesicle recycling? So when you have to recycle these vesicles, what role does AP2 actually play? Then the next question was whether the AP2 complex plays an exclusive role only in vesicle recycling, or is it also required somewhere else? And the third question was how is a defect in endocytosis linked to aberrant synaptic growth? So this question basically comes from the results that we got when we perturbed the function of AP2. So to tell you a bit more about the AP2 complex, so this has got four different subunits. The alpha and the beta are the two bigger subunits. You've got sigma 2, which is the medium subunit, and the smallest subunit here is the sigma 2. And this kind of holds together the whole complex. So what is interesting is that if you take away one of these subunits, the whole complex falls apart. Um, but there are studies in other model systems, particularly in the one in C. elegans, that shows that if you take apart this system, the alpha and the sigma can form a hemi-complex and can work together, and the beta and the sigma can actually form a hemi-complex and work together. So one of the things that we actually tested is whether that is true in Drosophila, but in Drosophila, this does not actually happen. So if you take out just one of these, everything else falls apart. So, uh, so what I uh, then did was basically try to understand what the function of the sigma 2 complex was. I mean, sigma 2 in putting together the complex was. So when I started off, we had a mutant in the lab, and that mutant had a synaptic morphology, which I'll come to in the next couple of slides. And since the morphology pretty much resembled a bunch of grapes, we call this phenotype as angur. Now, when I joined the lab, this mutant was not characterized. That is, we did not know what was the gene that was responsible for this particular phenotype. So what I then did was I used a deficiency mapping approach. So in flies, we have different deletions available, and we know the chromosomal locations of these deletions. So what you do is in standard genetics is you just take this particular allele and cross it with one of these lines, and you see whether they complement or not. So uh, the idea is if they do not complement, then that is probably the region where the particular gene is located. So in this case, uh, you can see that there are three different regions spanning this locus. And when you cross this particular mutant uh, with, you can see that the AP2 sigma gene is located here. That is common to like all these three deletions. So when you cross it with any of these three deletion lines, you will not get viable animals. So they will always be lethal and they will fail to complement, which means that your particular particular gene or particular allele lies somewhere in this region. So firstly, we do a broad kind of a screening where you have these bigger deletions, and then you roughly locate the region. So from here, we kind of located this particular region, which had like different candidates. You can see that there are like almost 10 different candidates out here. So the next step would be to take mutants from all these 10 to 11 genes and do a candidate gene screening. So when I did that, I found that a particular mutant or mutation in this locus, the AP2 sigma locus, did not complement Angur. So that is where we came to know that this particular allele is actually an allele of sigma 2 adaptin. So to further confirm if this was really the case, we looked at the transcripts, like the AP2 sigma transcript in the different combinations. And we found that compared to the control in all these mutants, the levels of the AP2 sigma transcript was severely reduced by many folds. And to functionally prove that it was indeed the case, what we did was in this particular mutant, when we put back AP2 sigma transgene, we were able to rescue the phenotype. And also when we knock down the gene, you see have a similar kind of effect. Now, how does this actually happen? I'm going to show you in the next slide. So in flies, we have a binary UAS GAL4 system. So this is actually borrowed from the yeast. So the yeast transcription factor is called GAL4, and that binds to something called as the upstream activated sequence. So there are two components to this. So you have a fly, say let's say this is a female fly. So you have the promoter sequence of a particular gene that is cloned upstream of the GAL4 encoding DNA. And in the other fly, you have the UAS sequence downstream to which is your particular gene of interest. Now, and this could be a transgene, this could be an RNAi construct. And so if you have an RNAi, you can use this to knock down your particular gene. So when you cross this, say, and if this promoter sequence is, say, a neuronal promoter, so you can achieve a tissue specific knockdown. That is, you knock down this particular gene only in, say, neurons. And then, of course, you see what is the consequence of that. So in the previous slide, what I showed you was something as actin 5C. Now, actin is an ubiquitous protein. So if you use an actin promoter and knock down this particular protein, you're basically knocking down AP2 sigma in the entire animal ubiquitously. And then you see what the phenotype is. So these are some of the genetics that is available. And then there is something called as FLP or flipase mediated recombination. Now we know that a recombination happens in meiosis, but you can induce uh, recombination to take place in mitosis, and that is with something called as the FRT flip method. And what you have here is a sequence called flippase recognition target FRT. So if you have this sequence in the chromosome, and then if you come with an enzyme that is called flippase that recognizes these sequences, it will induce a recombination event between the two FRT sites. And as a result, what you have is a series of clones wherein you have one clone that is a homozygous mutant, and you will have two heterozygotes, and you will have one which is a wild type. So this is particularly important when you have a mutation which is lethal, and I'm going to show you that in the next slide. So this particular mutant Angur was lethal. So you basically don't have a homozygous viable animal. So then the question is, if you don't have an animal in the first place, how do you actually study it? Because a dead man will not tell you the story. A dead man tells no tales, right? So, so the beauty of this genetic trick then helps you to study the function of this particular gene. So this is a wild type fly, and not a wild type. Actually, it's the FRT with the mutant Angur 7. And here in this case, we made clones in the eyes. So what happens here is that you've got the animal, which is viable, but the eyes are homozygous mutant for this particular allele. So that's the beauty. You have a fly which is uh, heterozygous, but the eyes of this particular animal is homozygous mutant. And then you basically can now study the function. So this is a bright field image. Then we wanted to study in greater details what really happens. So we looked in using a scanning electron microscope. Of course, this work was done in collaboration with Dr. Praveen Verma at NIPGR New Delhi. And you can see that the wild type has these 700 to 800 structures called omatidia. And these omatidia are separated by these bristles, which are like placed between each of these omatidia. Now, when you look at the mutant, you first of all see that the size is very much reduced. And you can also see in the inset that the morphology is very much awkward. You don't have those fine rounded structures that you see in the wild type. Furthermore, you don't really have those bristles that separates the structures. So that was an indication that something is really kind of wrong. And then just to make sure that it is this phenotype is because of the sigma 2 adaptin, when you provide sigma 2 adaptin back to the eyes in these animals, you see that you are able to partially rescue the phenotype. So we were interested in further understanding, like at the ultrastructural level, what really happens. And so this is a transmission electron micrograph. So you take a cross section of these omatidia, and what you see here are, so these are called as the rhabdomeres, and there are 8 such rhabdomeres, R1 to R8. Then you can see only 6 or 7 of them. So this is a wild type rhabdomere. If you look at the mutant again, you have absolutely nothing. So this is just like a hollow space here. So there are no rhabdomeres. Again, you put back the transgene, and you're able to rescue the phenotype. So that tells us 2 things. That is one, that it is indeed an allele of sigma 2 adaptin, because when you put back the gene, you're able to rescue the phenotype. And secondly, what I'm not showing you here is also we did some functional assays uh, using electroretinograms. That is, we tried to figure out what is the response of these flies to light, and we did find that they could not really respond to the light. So that again tells that there is a functional problem. And the reason you look at eyes in the first place is that, so you've got synaptic connections also in the eyes. So you have the retina, and then you have the lamina below that, and then you have the other structures that, and there are neurons connecting them. So this is a coarse way of actually first figuring out if there is a defect. And if there is a defect, then you can study further in the neurons. So this was our first study, and we found that, yes, indeed, there is actually a problem. And of course, this is what I already talked to you about. So now we wanted to look at the terminals, or the synaptic boutons. So I'm going to, in most of the slides, you will see this image, and that is just basically to guide you what we are actually looking at. So this is the entire larvae, this is the ventral cord, and the green one are the muscles, and there's something in red that is actually the neuromuscular junction, and this is exactly what you are seeing here. So this is the control. You have those nice varicosities. It's like a bead of string, very nicely arranged. But if you look at the mutant, what you'd actually see is that you have those much smaller structures, and they are a lot more kind of clumped together. And this actually resembles a bunch of grapes, and that's from where this name Angur is actually derived. So now you see that there is a definite morphological defect in these mutants. So we then quantified a few different parameters. That is, we counted the number of these rounded structures, and we did find that the mutants, all of them have significantly more number of these boutons. And of course, when you put back the transgene, you're able to rescue it. And we also quantified the area of each of these rounded structures, and we find that, of course, in the mutants, this is very much reduced, as is obvious from the image. And of course, when you put back the transgene, you're able to rescue the phenotype. So the message from this slide is that, of course, when you have a defect in endocytosis, when you have a defect in the AP2 adapter complex, you end up having a defect in the synaptic morphology. Now, we wanted to further look at what happens if you also knock down the other subunits. Now, this in the previous slide, what I've shown you is this is a mutant. So we wanted to see what happens you knock down the other subunits. Like, so I showed you in the previous slide that there are 4 different subunits: the alpha, the beta, the mu, and the sigma. So what happens if you knock down the other subunits, and we have a similar kind of a phenotype. So that is, we say that knockdown of any of the other AP2 subunits phenocopies the sigma 2 adaptin mutant phenotype. Furthermore, we also knock down an enzyme, that is PI4K3 alpha, that converts the PIP2 to PIP3 in the membrane, and we again have a similar kind of phenotype. Again, this is the quantification, the same kind of quantification, and you have a similar kind of result. This was performed at 2 different temperatures because the GAL4 is pretty much effective at higher temperatures. So we used the 2 different temperatures, and it is actually the case in both the temperatures. All right, so now what we wanted to then do was figure out if this is required specifically for the AP2 complex, or it is also required for other stuff. So again, this is the neuromuscular junction. We looked at the alpha adaptin, the beta adaptin levels, and we do find that they are drastically reduced. And then you are able to get back the levels when you put back the transgene. We also then looked at the levels of clathrin. And as I said, AP2 is a major adapter for clathrin, and we do find that clathrin is also reduced at the terminals. Further, we thought we should do some protein analysis to look at the total number of proteins. So this is a Western blot. And what you do see is that in the 2 mutants, you have almost nothing of alpha adaptin and beta adaptin. But for the clathrin, you do have a reduction. So it's almost like 50% reduction in this case. But you have some amount of clathrin left, and that tells you that when you don't have AP2, there are other mechanisms, that there are other proteins that can actually take care of clathrin stability. So there are other modes of endocytosis that is kind of still operating. And of course, alpha actin here is a loading control. So, so far what I've shown you is that the sigma 2 adaptin, the smallest subunit of the complex, is actually important for keeping the complex together, and it is also required for the stability of clathrin. So there's another structure that you have. You have those microtubules and microfilaments within the synaptic within the synapse. So you have the cell body, and then you have these tracks. These tracks are made up of microtubules and microfilaments, and they extend right towards the synaptic bouton. And why are these structures important? Because there's a lot of trafficking that takes place from the cell body to the synapse, or even like retrograde transport from the terminals back to the cell body. So there's a lot of communication that is happening here, and these microtubule tracks actually function in helping this transport. So, and again, if you look at some of the classical mutants, in this case, I'm showing you one of the mutants, which is called DAP160. Again, this is also a protein that is involved in endocytosis. So compared to the controls, when you look at these mutants, you see that there are kind of breaks. So this is a continuous structure. You have those rounded loops. So this is a labeling for a protein called as FUCH, MAB associated protein, and you find those wonderful loops in these control animals. But in the mutants, first of all, you have an interruption. So you can see that there are breaks in between. So these tracks, or the tubulin based cytoskeleton is not continuous. There is a break. And this is exactly why you also have defects in synaptic morphology. So what we wanted to then see was whether this was also the case with our mutants. And when we did a similar staining for the Angur mutant, we did find that there are towards the distal end, you have got these breaks. So if you see in the control, you have these looped structures, but in the mutants, you do get these breaks, which are like indicated with an arrow. And then again, of course, you put back the transgene, you're able to rescue the particular phenotype. So as a mode of quantification, what we did was we counted the number of boutons that have these kind of loops. And we found that in these mutants, we have a severe reduction in the number of loops compared to the control. And again, we were able to, of course, rescue the phenotype. So so far I've shown you what are the different defects that the sigma 2 adapted mutant has. So we then wanted to see at the functional level what really happens. So we then resorted to electrophysiology, and the way this experiment is done is that you have a recording electrode that is poked into the muscle, and then of course you record these traces. So these are the spontaneous vesicle fusion events. Every time a vesicle fuses, so I showed you in the previous diagrams that you have those synaptic boutons, you have the neurotransmitters that being released, and then of course there are receptors. So this is the neuromuscular junction. So the receptors are in the muscles. So when the receptors get those neurotransmitters, uh, they'll show some activity. So that activity is basically what is shown in these traces. And these are called as the spontaneous fusion events, or the miniature junction potentials, and that happens all the time. And so this is the kind of response that you get. But if you then take another stimulating pipette. So in this case, if you remove this brain, so the brain is basically sending the action potentials for the uh, muscles to depolarize. So in this case, you remove the brain and you take those nerve fibers in a stimulating pipette, and now you control the stimulation that you can actually give. So you basically inject current into these nerves, and then you see what is the response of the muscles. So when that happens, you get something like a big spike. And this is called as the evoked junction potential, wherein you have a synchronized vesicle fusion. So here, this is a very random event, but here this is a synchronized fusion event when you actually inject current, and then you see what is the response. So we did a similar kind of experiments with these mutants, and what we did find was that the spontaneous uh, miniature junction potential amplitude as well as the frequency was more in the mutants. And this was again similar to identical to the alpha adaptin RNAi knockdowns. So this was the minis, and then of course when you do the evoked synchronous fusion test, you have a similar kind of there is a reduction in the mutant and the alpha adaptin RNAi. Of course, we could rescue this by putting the transgene back. So again, we quantified these amplitudes, and what we did find was that there was a significant reduction in the amplitudes of the mutants or the knockdowns. And again, we could rescue back these parameters by putting the transgene. Then we looked at the quantal content. So quantal content is a ratio between the evoked potential divided by the mini amplitude. So when you do look at this ratio, you find that there is a significant reduction in the quantal content in all these mutants. And again, the rescue, you can bring it back. Now, this was performed by an electrophysiologist colleague of mine, Zishan. And what the quantal content actually tells us is, it tells you the number of vesicles that are being fused per action potential. So you have a rough idea of how many quanta of vesicles are actually fusing per action potential. And so we now know that there was a defect in the quantal content as well in these mutants. So we then went ahead and tried to figure out what really happens if you give a high frequency stimulus. So in one of the diagrams, I did show you that there are these synaptic vesicles. So if you give a high frequency 10 hertz stimulation, what happens is that you basically deplete all the synaptic vesicles. And then what happens is that uh, you would not really get any signal. Then you give a period of rest for some time. So when you give this period of rest, there is a refilling of the vesicles, and then you can kind of get back the same amplitude as you see in the starting with respect to the first action potential. So when you look at the mutant, or even for that matter, the RNAi, what happens is there is a gradual reduction in the amplitude over a period of time. And even if you give a period of rest, they are not able to recover back to the wild type levels. So you can see that these are the wild type levels, which they recover after the period of rest, but these in the mutant animals, you don't see any such recovery. And this is again similar in the case of when you knock down the alpha adaptin RNAi. And in the rescue, when you put back the transgene, you almost get back to the wild type levels. So this is what really happens. So we try to understand like what could be the actual reason behind that. And for that, we again collaborated with NCBS, and we did electron micrography of the synaptic boutons. And what you see here in cyan is the presynaptic part. You have those round vesicular structures. This dark one is a mitochondria here. And again, in the mutant, what you do see is that you have those bigger vesicular structures. You have something like a big thing bulging here. You have some bigger structures out here. So that kind of explains, if I go back to the previous slide again, you have those bigger amplitudes. So you have so now you can kind of appreciate what these bigger amplitudes mean. So these mean that you have got bigger vesicles, and they of course will have more neurotransmitters. So once they are released, you have these kind of bigger spikes. And also the fact that they would release more neurotransmitters, you also have these events happening more frequently. So that kind of explains what you see in the previous results. And so we do kind of understand now that this particular adapter complex, the AP2, could be actually involved in regenerating synaptic vesicles, probably from this kind of bigger structures. Now, when I say this kind of bigger structures, I'll come back to that in this slide. So the classical method of endocytosis, as I said, is you'll have these vesicles come and fuse, and they need to be regenerated back. And then, of course, they again reform the vesicles. Now, there are different models of by which these vesicles can actually fuse. So the one is called the kiss and run. So what happens in kiss and run is that you have these vesicles, they come and intermittently fuse with the presynaptic membrane. So they don't totally fuse, they just partially fuse, release the neurotransmitters, and they immediately are recycled back. And the classical way is wherein you have a vesicle, they come and totally fuse with the plasma membrane, and then of course they have to be recycled back. The third method is called as the ultra-fast endocytosis, wherein you have the fusion, and then the bulk of the membrane is kind of recycled back. They form an endosome, and from endosomes you again have these smaller vesicular structures which are being converted into the synaptic vesicles. And there's also a bulk endocytosis, wherein you have again bigger chunk of the plasma membrane that is being released, and then again you have to regenerate vesicles from these bigger structures. So what we kind of figured out from the previous set of data, that is the functional data and the electron microscopy data, was that probably the function of AP2 is not very critical when it comes to generating vesicles from the plasma membrane. But when it comes to generating vesicles from these kind of structures under high frequency stimulation, uh, it actually fails to do so, because there is a severe defect in AP2 function. So that also means that to make those vesicles from these endosome-like structures, you actually need clathrin and the AP2 complex, without which you cannot really make that. So uh, that's kind of what we figured out, and that is again consistent with the kind of results that were obtained or that is reported in some of the other studies, and they all talk about bulk endocytosis and the requirement of clathrin probably from this kind of structures, but not from the plasma membrane. So So to sum up the first part of the story that I've told you is that, sigma 2 adaptin regulates synaptic morphology. So if you mutate sigma 2 adaptin, you have some defect in the morphological phenotypes. And they not only facilitate basal synaptic transmission, but they are actually required for regenerating synaptic vesicles during high frequency nerve firing. They are required, I mean, the complex is required for the stability, for its stability and also for clathrin. And it regulates synaptic growth by controlling the microtubule cytoskeleton. Of course, this was published in 2016, and this is the reference. You could actually look back at it. So now coming to the second part of the same story. So as I've shown you before, like the mutant was called Angur because it had a aberrant synaptic morphology phenotype. And if you look back at some of the other mutants of the endocytic pathway, like endophyllin or synaptogenin, all of them have this similar kind of phenotype. So the question was, what actually goes wrong in these mutants, and what is the reason for this kind of aberrant synaptic phenotype? So again, that's what the question was. You have this beautiful control animal. There's all synaptic boutons are like arranged in a beautiful beads on a string. And here in this case, you can see that that is totally disturbed, and you have the smaller structures and this kind of a weird phenotype. So the question was, why is this happening? So to look at that, what we wanted to first look at was the growth signaling pathway. So this is not as simple as it looks here. I have kind of made it oversimplistic, but there's a lot of crosstalk that is happening, and there are more than just these two pathways. But for the sake of this presentation, I've just tried to show you two pathways. And one is the BMP signaling pathway, and the other is the MAP kinase pathway. So in the BMP signaling pathway, what happens is you've got MAD, which is actually phosphorylated, and then it forms a complex with COSMAD, which is called media, and this is then transported from the nerve terminals to the nucleus. So that's a retrograde transport. And then, of course, once in the nucleus, it activates the transcription factors, and then you have various growth promoting genes that are being transcribed. On the other hand, you have a MAP triple kinase pathway, and that is, there is a protein called Valenda that activates a Jun N-terminal kinase that activates transcription factor FOS in the nucleus, and that leads to the uh, expression of your other growth promoting genes. And there is this E3 ubiquitin ligase called HiWARE, which is at the intersection of these two pathways, and apparently it controls both the signaling pathways, because Valenda is a substrate for the HiWARE. So what it does, it sequesters Valenda. So if it basically degrades Valenda, you have this branch of the pathway that is kind of being controlled or kind of being silenced. So if you don't have HiWARE, or if you look at a HiWARE mutant, you have more levels of Valenda. And if you have more levels of Valenda, this particular pathway then gets hyperactivated. On the other hand, HiWARE also is known to interact with this particular protein called media. So if HiWARE binds to media, the MAD is basically free. So what happens is MAD cannot form a complex with media, and if MAD doesn't form a complex with media, you cannot activate the downstream signaling pathway that activates growth promoting genes. So HiWARE mutants have aberrant synaptic growth, and that is already documented. So we then wanted to look at first the BMP pathway, and the way to look at it is like we wanted to look at the levels of phosphorylated form of MAD. Again, you're looking at the neuromuscular junction here. In the control, you really don't see any of the staining for MAD, but if you look at the Angur mutant, you see that the levels of MAD are really high in these synaptic terminals. Again, when you put back the transgene, you're able to get back to the control levels. So for sure, the BMP signaling is upregulated in this particular mutant. Then we also wanted to see what happens to the other branch of the pathway. So in this case, you are looking at the ventral nerve cord. So this is this portion of the larva. And again, what you see here is in the mutant, you have those uh, staining pattern for Vallenda, which is really, really high. And when you put back uh, AP2 sigma transgene, you kind of get back to the control levels, or even if you put back a high wire, you get back to the control levels. So that shows that, Vallenda is also high in these mutants, and that's why the MAP kinase signaling pathway is also getting activated. So both the pathways apparently seem to be activated in these mutants. And what we then next did was we looked at the other downstream factors, as I showed you in this slide. So these are the different proteins that are part of this pathway. And if you want to really understand the function in a pathway, you then try to basically uh, manipulate some of these players, and then you see what really happens. So we were talking of these branch of the pathway, and then the idea was if you can say block either JNK or block either FOS, what really happens to the growth. And that is exactly what we did. So we expressed a dominant negative form of the JNK and the dominant negative form of FOS. So that is essentially you are disrupting the function of these two particular proteins. And what we do see here is that compared to the mutant terminals, which are very much uh, clustered here, you see that there is some kind of a partial rescue, though the rescue is not 100%, but you definitely see some kind of rescue. And again, this is the quantification. You see in the mutant that there's very high number of boutons, but in the two rescue cases, you see that there is a significant amount of improvement when you compared it to the rescue animals. And of course, this work is presently being carried on by Manish Dwivedi and Isar Bhopal. So, so to sum up the second part, what I've shown you is that the sigma 2 adaptin regulates synaptic morphology by modulating the BMP pathway. So there are more experiments that are happening right now to understand how exactly the BMP modules are regulated. So there are these BMP receptors which are in the presynaptic membrane, and then of course we're talking of endocytosis. So there is a good possibility that these receptors are not being endocytosed, and that could be one of the reasons why the BMP pathway is hyperactivated. And what I've shown you here is WEND or Vallenda is actually upregulated in these mutants, and that leads to the activation of the transcription factor FOS, and in turn the MAP kinase pathway. So with that, I think I'll move on to the third part. And okay, before going that, I began with asking these three questions, right? So what was the role of AP2 in presynaptic vesicle recycling? And yes, we did find out that it facilitates basal synaptic transmission, but then has a minimal role in synaptic vesicle retrieval at the Drosophila neuromuscular junction. The second question was, what does it do? Or does it have an exclusive role in vesicle recycling, or is it required elsewhere? So the results indicate that it is absolutely required for regenerating synaptic vesicles under high frequency nerve firing, and also for stabilizing clathrin. And thirdly, how is the defect in endocytosis linked to aberrant NMJ growth? And we did find that defective endocytosis perturbs growth signaling pathways, resulting in NMJ, aberrant NMJ growth. And of course, the two pathways under investigation right now are the BMP and the MAP kinase pathways. So that kind of sums up my PhD thesis work. And now I'll just switch gears and move on to another interesting study that also I did during my stay at ISER Bhopal. And this is more related to diseases. And so this is something to do with amyotrophic lateral sclerosis. And I'm sure this image looks very familiar. So this is the ice bucket challenge that kind of went viral, I think around 2014, 2015. And this was done to raise a lot of money, and that was uh, then channeled to do research in ALS. So what really is this ALS, and how does it actually affect the muscles? So I showed you most of the slides previously where we looked at the neuromuscular junction, and you have those neurons that have those long axons, and they send their projections and they innervate the muscles, right? So what happens in these diseased conditions? So in the normal condition, you have a healthy nerve and then it innervates the muscles and it's functional as it's evident from uh, uh, the electrophysiology recordings. You see that absolutely normal. But what happens in amyotrophic lateral sclerosis is that first of all, you have damaged nerve. You don't really have the continuity. In most of the cases, you will have degeneration happening towards the terminals. They apparently don't innervate the muscles that well. And so what happens is the muscle loses contact with these neurons. And so functionally, uh, I mean, they're not able to respond to the stimuli. So this manifests in the fact that uh, you have muscle wasting and sometimes you're not able to say hold an object properly. You just don't have the stimulus to perform that motor activity. And so this is in brief what happens in ALS. Now, the next important thing is how does it actually happen or why does it actually happen? So ALS mechanisms are very varied. You have got different mechanisms. A few of them are listed here. You've got sometimes you have disturbances in protein quality control. The protein gets degraded or it fails to get targeted to the endosome, lysosome, or autophagosome. So you have got uh, uh, proteins which should actually be degraded, but they are not. So they tend to remain around at the synapses, and then that kind of hinders the whole process of synaptic communication. Sometimes what happens is you have the microglia which are hyperactivated, so they release a lot of these free radicals or reactive oxygen species, and they are again detrimental to the neurons and they result in cell death. You have these other problems where there could be defects in the lactate transporters. You don't have enough energy that is being that is reaching the synapses, and so again they kind of die out in time. Sometimes what also happens is that you have neurotransmitters that are being released. Now the neurotransmitters that are not being used up need to be recycled back, but sometimes due to defects in the recycling machinery, you have the neurotransmitters that remain in the synaptic cleft for a longer time, and that results in excitotoxicity, and that again kills the neurons. There could be defect in the cytoskeletal proteins. So I showed you previously that you have those microtubules and microfilaments that runs along these axons. So if there is some kind of a defect in those cytoskeletal proteins like dynein or dynactin, again, you don't have proper trafficking taking place, and then this kind of affects the motor neurons. And lastly, it can also be due to disturbance in RNA metabolism. So there could be problems with the RNA binding proteins which are somehow getting sequestered and they are not able to uh, bind to the target proteins, and then you either lead to less translation of the target proteins, or it could be that some proteins are highly translated at the synapses and that lead to this kind of defect. So as you can see, like there are varied reasons for ALS to actually happen, and it's not just familial. Like 10% of the cases are familial, that is, it's hereditary, but 90 to 95% cases are sporadic. So even if you don't have a family history of ALS, uh, you still have a chance of developing ALS. So that is a serious issue. Um, then of course there have been many genes which have been implicated for ALS, and so they are grouped into different subcategories. You can see that there are RNA processing genes. There is something called to do with glutamate excitotoxicity, oxidative stress, protein degradation, chromatin remodeling, endosomal trafficking, mitochondrial dysfunction. So there are many more. So you can see that there is a very wide spectrum that is associated with ALS. So what I'm going to take you through in the next couple of slides is our work with this particular protein called Senataxin or SETX. So SETX is RNA binding protein, and this is the human protein, and you've got different domains like a protein interaction domain, ATP, JTAB binding, motif A, and then you of course have the most important DNA RNA helicase domain. So this is the domain with which it uh, uh, binds to the different RNAs and then probably regulates its function. So when we look back the literature, there are from different studies across patient samples across the world, um, there were a couple of mutants that have been identified, like the T3I, R2136H, like you have an arginine to histidine mutation at 2136H, and then you have a 1363. That is, there is a stop codon that is being introduced at the position 1363. So what happens is you get a truncated protein, or you also have something like a threonine to isoleucine at the third position. So there are different mutants, and these have all been like reported in human populations. So what we then did to study the function of this protein in fruit flies, uh, we made different transgenes and we expressed this particular protein in the Drosophila neurons. And we then wanted to see what is the consequence of expressing these uh, human protein in flies. And what we did find was if you express the wild type or the two other mutant versions in Drosophila neurons, you find that it leads to NMJ structured plasticity. What I mean by that is that you've got more number of synaptic boutons, and you can see that there is a parent bouton here which is surrounded by these small structures called as satellite boutons, and this is the case with both the mutants as well. So the wild type as well as the control, as well as the mutant forms of these particular overexpressing animals show increase in the bouton number and the bouton area. So this a similar kind of quantifications that I've shown you previously. And yeah, so this is something that we figured out. And then we wanted to next see like whether that is because of a perturbation of any of the signaling pathways. And here again, you're looking at the neuromuscular junction. And again, the two most studied pathways, as I said previously, are the BMP and the MAP kinase pathway. When we looked at uh, the the PMAD signals in these animals, we saw that compared to the controls, the signals were higher in these wild type or the mutant animals. And again, in the wild type and the mutant, you have the protein called HIVER, the E3 ubiquitin ligase, which has actually gone down. So this shows that when you express this particular protein in Drosophila, what happens is you perturb the signaling pathways, mainly the BMP and the MAP kinase pathway, and that results in aberrant synaptic growth. Um, what we also did was we expressed the truncated protein, and we expressed the truncated protein in muscles. And this result was really interesting. So what you see here is the muscle that is shown here, the round structures, the nuclei, the muscle nuclei, which is labeled with DAPI. And then you have this protein called ELAV. So this is an RNA binding protein. And so this is actually a protein that is present in neurons. So it is not supposed to be present in the muscles. As you see in the control animals, you don't have any signal for ELAV. But when you express this truncated form of protein in the muscles, you see that ELAV expression is very much strengthened in this case. And with this, what happens is we kind of proposed a model wherein human Senataxin somehow is relieved of the repression. So in the muscles, you have the ELAV mRNA that is somehow translationally repressed. But when you express this human Senataxin, what it probably does is it kind of interacts with other proteins that are known to repress the translation of ELAV in the muscles. That interaction is disrupted, and then you have ELAV being kind of expressed. And this is an interesting finding because as I said, the RNA binding proteins and one of the key causes of Senataxin dysfunction. So, and we probably, though this is not in the human or the human cell lines or vertebrate system, yet I would say that in a heterologous system, this is an important finding because you kind of show for the first time that uh, you perturb the translational machinery, and that could be one of the reasons why the the patients having this kind of mutation, which removes the DNA RNA helicase domain, kind of fails to work properly. So to sum up what I've shown you in the Senataxin story is that the expression of the human Senataxin in Drosophila motor neurons induces some morphological plasticity. And then, of course, you have the human Senataxin uh, when expressed in motor neurons alters the BMP or the MAP kinase signaling pathways. And when you have the truncated form of Senataxin expressed in muscles, it alters the translational profile of ELAV, that is an RNA binding protein, and actually it might be altering many more other RNA binding proteins. And again, these two stories are published, and this is the reference for that. So with this, I would like to just have one slide telling you what I'm doing in my postdoctoral work. So the previous work, what I've shown you today is all from my doctoral work. And um, as was pointed out previously in the introduction that in my postdoc, I'm looking at the postsynapse. So you have those receptors that are in the postsynaptic membrane, or say for the Drosophila NMJ, it's the muscles. So they need to be, they are synthesized, they are translated, they have to go through the ER and Golgi network, and then they have to be, you know, these receptors have to form functional tetramers, they have to be trafficked or to the surface of the muscle, then they have to be kind of clustered and sent to these synapses, and then that's where they are kind of functional. So the study basically involves what are the different mechanisms by which these receptors are stabilized, what are the different mechanisms by which they are trafficked to the synapses, and how does the clustering actually take place. And there are interesting studies where you found auxiliary subunit proteins that help in the surface delivery and the clustering of these molecules. Of course, that story is probably for another day, but that is something that I'm actively looking at presently and trying to understand the postsynaptic differentiation and how you actually sculpt synapse, and more from the postsynaptic side. So with this, I would like to acknowledge the people who've helped me during my PhD and all who are helping me now in my postdoc. So from ICER Bhopal, my mentor, Dr. Vimlesh Kumar, and this is the bunch of people who've been associated with the work. Um, Sishan, Abdul, Manish, Bhagwan, Asim. So they've been mostly involved with the sigma to adaptin work. Sri Krishna, Namrata, and C have been involved with the Senataxin work. Abhinandan was recently involved with a part of the sigma to adaptin study. Akil is the person who takes care of the flies and make the food. So he's an important resource. And most importantly, Dr. Vimlesh Kumar is a wonderful mentor. And well, I've spent like 6.5, 7 years in Bhopal. So it was a learning experience working with him and working with fellow colleagues. And at the National Institutes of Health, I have a wonderful mentor in Dr. Ella Serpe. She's very encouraging and very supportive. And you've got a bunch of colleagues. They're all postdocs: Rosario, Tahi, Thol, Lindsay, Wenjay, and Tom Brody. Tom Brody is here. So he's a veteran, and he is also the one who maintains a website called the Interactive Fly. Uh, and well, people familiar with Drosophila would definitely know him. Uh, Peter, of course, takes care of the lab, and uh, the current funding is from the NIH intramural funds, and for my PhD work, I was funded by the Ministry of Human Resource and Development, the Department of Biotechnology, the Department of Science and Technology, and the ICERB intramural funds for the experiments. So Thank you so much for your patience and for listening. And if you have any questions, I'll be very happy to answer.
Thank you, Soumitra. Now the session is open for questions. Participants, if you have any questions, you can post it in the chat box. Mhm. Okay, I think now at present there are no questions. Okay, maybe later. Okay, yeah, there is a question coming. So he's actually talking about the particular slide device. In slide
Okay.
21, can you see that?
I can see the question. He says slide 21 alpha adaptin
Yeah.
RNAi current is not going back to the basal level as like the other variants. Why is this happening? So this is an RNA lane, and it could actually have possible problems. The knockdown may not be as efficient. So yeah, that could be an essential. That could be a reason, I would say. The other ones are the mutants. So you have it like the mutant is like it's a mutant in the whole animal is a mutant, but if you look at the alpha adaptin RNAi, you're just knocking it down in the neurons. So there could be other things happening from the muscle side or from the glia, and also the knockdown could not be 100% efficient. So there could be that kind of a problem. Yeah.
Any more questions? Yeah. And one more question came, Soumitra. Has there been progress in studying the genetic aspects of diseases?
Yeah, I mean, using the fruit fly, yes, there have been many studies using human diseases, because as I said, like 70% of the diseases that occurs in humans, you have a particular homologue in flies. And the advantage of using flies is that in humans, you might have different redundant genes. So if you knock down one, or if there is a mutation in one, the other proteins can actually take over. But in flies, most of the cases you just have one particular gene, so you usually get a phenotype when you mutate that. So yes, that happens. And then he's asking, rather than just the superficial causes like defective proteins. Okay, so this was just one example that I showed you today with Senataxin. So this is known to have a problem with RNA binding, and then you have this kind of things. But you have other diseases in which people are actually figuring out the mechanistic reasons behind what is actually happening. So there's a lot of disease work that is actually going on. So and since as I said, like there are functional homologues, it's very easy to actually figure out the mechanisms. Yeah, but one has to be very careful, because the disease work is it's demanding, or it's challenging, I should say, and it's easy to come out to have wrong conclusions. So that is one major problem, I would say, in the field. So one has to be very critical with, when you are studying diseases. Yeah. Does that kind of answer your question? I hope it does.
Okay. So thank you so much, Soumitra, for sharing your informative talk with us. Okay, started with the basic, with the diagrammatic representation and all. It was so nice. Okay, the way you presented, taken us into your doctoral discussion, and then later your postdoc thing also you have little bit touched. So it was very nice, very informative session. Yeah.
Thank you.
Yeah. So we will conclude now with the vote of thanks.
Okay.
Okay. Respected Principal, Dr. E. Jerome Xavier, Indian Academy Degree College, Autonomous. Dr. Prakash, Scientist in University of Iowa. Dr. Soumitra, Scientist in National Institute of Health. Members of faculty, and all the student participants. A very good afternoon. It is 12 now. I deem it a great honor to propose the vote of thanks. First of all, I would like to thank our chairman, sir, Dr. T. Somashekar, Indian Academy Degree College, Indian Academy Group of Institutions, and our secretary, ma'am, Mrs. T. Bharati, Indian Academy Education Trust, for their constant support. I would also like to thank our COO, Dr. Srinidhi K. Parthasarthi, for his support. I would like to extend my heartfelt thanks to our beloved principal, Dr. E. Jerome Xavier, Indian Academy Degree College Autonomous, for his continuous support and encouragement. I would also like to extend my thanks to our resource persons, Dr. Prakash and Dr. Soumitra, for making this event a memorable and a successful one. And let me thank all the participants for their presence and active participation. I would also like to thank our HOD ma'am, Dr. Prashanti, for her help and support. And all this can't happen without a teamwork. So we had a very nice team, including Professor Dilshad Begum, Professor Malati, and a special thanks to Mr. Chandan, the head of the system admin team, for his sincere efforts in helping us in arranging the webinar. I also thank all the technicians for their help. Once again, thank you all of you.